Hello, Jai.
Hello.
I think it is time to start.
Yes. Just few more minutes, sir. Hello, Deshmukh sir, are you there? Hello, Sujit sir.
Yes, yes. Am I audible?
Loud and clear.
Okay, thank you.
Deshmukh sir will give you the co-host, Sujit sir. So you will be
Okay.
able to share your PPT.
Sure, sure. Let me know, then I can share my screen then.
Sure.
Sir started.
Hello, Prakash sir. How are you?
Hello, namaste.
Namaste, sir.
Who is your guide? Is
Sorry,
it
sir.
Yes, sir, Joshi?
Sir, am I audible, sir?
Yes,
Yeah,
sir.
yeah.
Yeah,
Who is
my
your
guy,
guide?
my supervisor is Dr. Mishra, Dr. BK Mishra from Nehu.
Okay, okay.
Very nice.
And my joint supervisor is from Anand, Gujarat only, Dr. Subodh Hathi.
Oh, you work with him? He's
Yes,
my good
yes.
friend.
Yes, I know. I have been reading a lot of papers, sir. I have been
Wonderful.
seeing you in conferences also. A lot of conferences I've seen you. I've been reading your papers, and Hathi sir has told me about you. Dr. JB Prajapati also.
Okay, okay. So you are part of a Sasnet also.
Yes, sir. I'm a member of SASNET also.
Wonderful. Okay, I'm looking forward to hear you.
Sure, sir. Sure.
Sujit, where did you do your PhD work?
Sir, from Northeastern Hill University, Shillong. Yes.
Very good.
And
And now
along with, so now I am in BIRAC. I'm working in BIRAC.
in Delhi?
Yes, sir, in Delhi, BIRAC-DBT.
Oh, very good. Very good. Very good. I had many projects from BIRAC.
Okay, great, sir. So I'm working here in the technical and scientific division.
Very good. Very good. You are a doctor now.
Yes, sir. Right
So
now I am.
congrats.
Thank you, sir.
Congrats.
Thank you.
So why don't you start now? Because people will join in.
Sure,
Yes,
sir.
yes. Just Deshmukh sir is there, na? So once you give him the course, we can immediately start.
Actually, we should maintain the time.
Yes. But we are also looking for participants. But yes, that is absolutely correct, Swanjit sir.
See, 5 minutes is okay, but not more than 5 minutes.
Yes, totally makes sense.
You call him. Call Deshmukh ji.
So good morning, everybody. Am I audible?
Yes, you are.
Okay. I hope everyone is okay. So we are just starting a lecture in few seconds. Sorry for the little delay. There is just some technical issue.
Okay. If it is technical, it is all right. But otherwise, you should start in 5 minutes time.
All right, perfect. So good morning, one and all. Today's our speaker is Dr. Sujit Das. And first of all, I would like to say MSI Gujarat President Dr. Minal Trivedi has given her regards. Ma'am could not join because she is at the hospital. Some relative of her is not okay, so she is there. But she has given her regards. And also, I would like to say Dr. Arvind Deshmukh sir has also given his regards. There is some issue on his end, so he is not able to speak. So in the interest of time, we would immediately start. Today's our speaker is Dr. Sujit Das. He is currently associated with BIRAC as a technical officer, Government of India. Before that, Sujit Das sir was given a Young Scientist Award from the Microbiology Society India only. Dr. Sujit has done his PhD from Northeastern Hill University, Shillong, Meghalaya, India. Dr. Sujit has done his MSc Microbiology from Assam University and his BSc from University of Burdwan, West Bengal, India. Other than that, he has worked as a GRF and SRF in Government of India funded various projects, and he has also worked as a research fellow associated with Government of India. Dr. Sujit has more than 24 very nice publications, I must admit. It looks very good. Other than that, Dr. Sujit has published 4 book chapters, and he has also published 2 manuals. Dr. Sujit has received more than 12 awards for his scientific work, and as a research person, he has joined and worked with many webinars and series. Dr. Sujit has given more than 12 presentations in various national and international conferences. And Dr. Sujit is a life member of Microbiologists Society India. Now, in the interest of time, I would ask Dr. Sujit that please start your lecture.
Thank you, Mr. Jay, for the introduction.
Thank you.
I'll share my screen.
Sure. Let me know if you can.
Mr. Jay, it's coming like host disabled participant screen sharing. Can you please help on this?
Sure. Just a moment. Just a moment. Deshmukh sir, if you can give host to Dr. Sujit.
Hello. Hello.
Hello, sir.
Yes, sir.
Can you hear me?
Yes, we can hear you.
Okay. So
Yes.
first, Prathamesh, because he is a host now, Prathamesh, make Sujit Das as a co-host.
Right.
And Swarnajit Singh sir will talk on my behalf. There is some issue with my mobile.
No worries, sir. No worries.
Please, Swarnajit sir. Swarnajit sir, please start.
Sir, please, I request Dr. Jay, can you, I'm unable to share my screen because like the same thing is coming.
1 minute. Sujit Das, 1
Just
minute.
a moment.
Oh,
Please.
okay, sir. Okay. Sure.
So, Swarna Singh sir will proceed. Swarna Singh sir.
Okay, sir.
Hello, Swarnajit sir.
Yes.
Please proceed. Give introduction of the
Uh,
talk.
Hello.
Yes, we can hear you, sir.
Can you hear me?
Yes. Loud and
Dr. Deshmukh just called me and asked me to say a few words on his behalf.
Please, sir. Please proceed.
Can you see me? Can you hear me?
Yes, very much.
All right. I'm very happy that we have another PhD seminar, and this time from the Northeast, from the Northeastern Hill University, Shillong. You'll be happy to know that I did my BSc from Nihu, but I was in Kohima, Nagaland, when I did my BSc. So I know the region very, very well. It's an extremely beautiful region, the Northeast. And I did my PhD from Jorhat, Regional Research Laboratory, Jorhat. That is also in Assam, in the Northeast. So when I read the topic, Lactobacillus, from traditional food of Meghalaya, I was very, very happy. And I would look forward to listen to the interesting seminar from the Northeast India. So I, on behalf of Dr. Deshmukh and the Microbiologists Society of India, welcome you to deliver the seminar. Thank you.
Thank you so much, sir.
Sujit sir, you may start now.
Yes, I'm. Please let me know if my screen is visible.
Yes.
Yes, sir.
Okay.
Please proceed.
Thank you. Thank you, Dr. Sonajit Singh. Before starting, I'd like to thank the Microbiology Society India, chapter Gujarat, for giving me this opportunity for presenting my research topic. And I'm really thankful to the All India President, Dr. Deshmukh, the State President of Gujarat chapter, Dr. Trivedi, and Mr. Jatiya, the Organizing Secretary of Microbiology Society India, for taking up all the pain and giving me the chance. So my research, as you have already sir has said, like in isolation and characterization of lactobacillus strains from traditional foods of Meghalaya for the development of probiotic soy yogurt. So my supervisor was Dr. B.K. Mishra from New Shillong, along with the joint supervision of Dr. Subodh from Anand Agriculture University in Anand. So before jumping into my findings, very quickly I will elaborate why particularly I chose this topic. So like we know, the probiotics, it's a term we all know it's from Greek literature, pro bios, which means for life, which is as opposed to as antibiotics, which means against life. And the first pioneer was Dr. Eli Mechnikov. He was a Russian researcher, Nobel laureate in 1908, who first witnessed the beneficial effects of probiotic microorganisms. And since there are more than bacterial cells in the human cells in the body, we must take care of it for proper functioning of our daily life and gut microflora as well. So this is the area of concern, lactose intolerance, which we know it is a general condition occurring due to the deficiency in lactase enzyme in the small intestine that is responsible for breaking down this disaccharide lactose into its individual components, galactose and glucose. And when this is not present or is present in scant amount, so this leads to cause of bloating and primary lactose malabsorption, as you can see in the animation to the right hand side. So to counter this, we thought of choosing soya milk because it might be considered as an economic alternative for bovine milk, as it is lactose free as well as galactose free. So this is the statement of the problem, the exact reason why we have done this. So like most of the people in Northeast region, especially in Meghalaya, lactose intolerant. They're unable to digest bovine milk, and they suffer from milk allergies. That's why the dairy industry is not well established in Meghalaya as well as in most of the northeastern parts. So fermented soy milk are the perfect alternatives for the lactose intolerant population. And provided that soy milk is also galactose free, it can be also considered as a cure for infants suffering from galactosemia who are unable to absorb their mother's breast milk. So a diet almost similar to that of bovine milk can be given by soy products. Keeping this in mind, we have framed the objectives like to isolate, screen and characterize lactic acid bacteria from traditional fermented foods from locally available from Meghalaya, to evaluate the technological and the probiotic attributes of the selected lactic acid bacteria, to develop probiotic soy yogurt with selected isolates, their combination, and to assess the probiotic properties, and to study the shelf life of probiotic soy yogurt by sensory evaluation under refrigeration conditions, followed by animal studies. So these are a few of the fermented foods from which I have isolated the bacteria: the fermented fish, fermented bamboo shoots, fermented soybean. We basically chose lactic acid bacteria only from these fermented foods because they execute a pivotal role in long-term preservation and production of nourishing fermented foods. So the diverse group of homofermentative and heterofermentative lactic acid bacteria will help in competing with the spoilage organisms. We have done the physicochemical characterization followed by the molecular characterization of the lactobacillus isolates we obtained from these fermented foods, followed by 16S RNA amplification and molecular phylogenetic analysis of the lactobacillus isolates. So we have collected around a total of 75 fermented food and beverage samples, out of which 34 we found lactobacillus species. But after molecular characterization, we took 8 forward, 8 best lactobacillus species followed based on the results of the Sankey sequencing. So these are the different probiotic attributes which we have studied, like alpha galactose activity, beta glucosidase activity, exopolysaccharide production, antimicrobial activity, tolerance to gastric juices, intestinal juices, antioxidative activity, autoaggregation, coaggregation, cell surface hydrophobicity, antibiotic susceptibility, tolerance to lower acidic conditions, bile salts, bile salt hydrolysis activity, proteolytic activity, and cholesterol activity. So these are the basic criteria where we have to analyze whether our bacteria are showing the particular probiotic attributes or not to claim it as a probiotic microorganism or with rich probiotic potentiality. Consumption of soya products is basically limited due to its objectionable beanie flavor and flatulence when consumed in large amounts. So the levels of oligosaccharide in soya products could be reduced by using this lactobacillus isolates, because it is the utilization of microorganisms possessing to different enzyme activities and supplemented with different nutritional factors. Is my slide moving?
Yes, yes, please proceed.
I'm unable to go. Just 1 second. It's not moving.
What happened? You cannot move
No,
forward or backward?
it was going fine till now, but I'm unable to move the slides. Just 1 second.
Sure.
I'll share again.
Sure.
I'm so sorry for this glitch.
No worries, sir, please.
Oh, this is not. Is this visible, Mr. Jay?
Yes, yes.
Yes, yes.
Okay.
Now it is fine.
I'm so sorry. It stopped moving. I don't know why.
No worries. No worries. Please proceed.
So yeah. So next, coming to the slide where I left off, the evolution of growth behavior of lactobacillus isolates in soya milk with nutritional factors. We used inulin skim milk powder and whey protein concentrate to check the efficacy of these prebiotics. So to make it as a symbiotic product. The reason for applying this prebiotics was to use it as a substrate for the probiotic cultures, so that it triggers fermentation in large intestine. So because few of the lactic acid bacteria, they have a low growth rate in soya milk due to the absence of lactose sugar. So we thought of supplementing this with different nutrition supplements to check the efficacy. So out of which iluminil proved to be the better amongst all. These are few of the proteolytic activity, beta glucosidase activity, and beta galactose activity we have done for the isolates, which showed on different levels of supplementation with inulin, skim milk powder, and whey protein concentrate. So out of all, again, inulin proved to be the better amongst all, to be used as a prebiotic along with our product. So based on the technological and probiotic attributes, the isolates, Lactose plantarum, 2 strains of fermentum, and 1 rhamnosus were further checked for efficacy in the production of B vitamins, vitamin B2, B9, B12, and organic acids production, along with biotransformation of soya solvents before the development of our final product. So the B vitamins or organic acid producing strains of lactobacillus could be considered as a primary factor to be used in the development of novel fermented foods, and they're more economically sound to be used rather than fortified with pseudovitamins that are synthesized chemically. So as you can see in the HPLC chromatograms in below, the production of B2, B12, and B9 by the lactobacillus isolates, followed by the production of lactate, acetate, and butyrate, accordingly by the lactobacillus isolates, and successful biotransformation of soya glycones during soya milk fermentation. As you can see in the image, the pictures of before fermentation, the production of daidzein and genistein, and after fermentation, the increase in quantity of the daidzein and genistein. Like after soya ingestion, the absorption of isoflavone glucosides in small intestine is very poor due to their hydrophilic nature. So the glycosides that are not absorbed in the small intestine pass to the colon, and during that process, if any probiotic microorganisms are present, they prepare for hydrolysis due to their beta glucosidase activity for releasing the free I-glycans as shown in the HPLC chromatogram. Next, we have standardized for the development of soy yogurt using different combinations of flavors, out of which white rose was considered was preferred by the sensory panelists. So we chose this particular white rose flavored yogurt to carry forward for our study, storage life study, and animal trials as well. So the different batches of flavored soy yogurt for standardization was done, out of which white rose flavored soy yogurt was used, followed by sensory study till 10 days, followed by the physicochemical and the microbial analysis. And also we have checked the biofunctional attributes of the rose flavored soy yogurt during the storage study, which included ACE inhibition activity, antimicrobial activity, total antioxidant activity, total polyphenol activity, and biotransformation of soy flavons. So these are the biofunctional attributes of functional soy yogurt. So again, the white rose flavored soy yogurt showed the highest ACE inhibition activity on day 4 of the storage study. Similarly, the anti-nutrients, the polyphenol content reduced till 10 days of storage study. And again, antioxidant activity was witnessed highest on day 4. This antimicrobial activity was also was increasing till day 10, but day 4 was considered as optimum for this as well. And this is again the biotransformation of soy isoflavones from the freshly obtained soy yogurt. The first image is before fermentation, the amount of daidzein and genistein, and after fermentation, the rise in the genistein and daidzein. So based on this, we have done experimental design, and for the antibiotic associated diarrhea, we chose 36 Vista rats, which were divided into 6 groups, each containing 6 rats. Since antibiotic associated diarrhea develops from the disturbance in the commensal gut microbiota due to excess antibiotic therapy, administration of this probiotic based food will be a logical therapeutic approach to regulate the gut microbiota. Thinking of this, we divided this Vista rats into 6 groups. The first group was normal control test group, where the untreated animals were served with normal soybean corn diet. Nextly was the disease control group, was administered with the antibiotic mixture using clindamycin, ampicillin, and streptomycin. Nextly, the standard group was administered with loperamide. Test group A1 was administered with functional soy yogurt. Test group A2 was administered with Lactobacillus plantarum and fermentum combinations. Test group A3 was administered with Lactobacillus fermentum and Rhamnosus combinations. So following the pre-challenge with antibiotic mixture for 7 days during the treatment period, the antibiotic acid diarrheal rats from each of the test group was orally gavaged till day 15 with 1 ml of each of the above mentioned samples twice a day. So this is the body weight parameter we have checked. So for investigating the metabolic changes, however, the body weight of the normal control group showed no basic alteration till the 15th of the study, and slight decrease was observed in the rest of the test groups from the 5th day onwards. This is the fecal consistency out of all groups: normal control, which was fed with the normal diet, and test group A1, which was fed with the soy yogurt, was comparatively least affected by the antibiotic associated diarrhea induction, based on the scores you can see in the graph. And on the last day of the study, the test groups A1 and the standard showed dry and normal consistency of feces with well-maintained and brown color of the fecal consistency score of 1, presuming that the antibiotic acid diarrhea was resolved in normal control as well as in the group fed with soy yogurt and standard drug loperamide. So here this is the fecal water content. The fecal water content also substantially increased in all of the antibiotic associated diarrhea from day 0 to day 1. However, the significant reduction was observed in group A1, A2, and standard drug. So mostly the reduction was observed in the one fed with test group A1 containing the soy yogurt. Nextly is the microbial analysis of the pooled fecal samples. Other than the normal control group, rest all of the 5 groups show significant decrease in the pooled fecal microbiota on the 5th day in case of lactobacilli and yeast cells. However, from 10th day onwards, maximum increase in lactobacilli was reported in group A1 fed with the soy yogurt. Nextly, coming to the hematological analysis, these are the blood parameters were considered as one of the key indicators in diagnosing actual and physiological status of the antibiotic associated diarrhea rats, an increase in the hemoglobin, hematocrit, mean cell hemoglobin concentration, mean corpuscular volume, and mean corpuscular hemoglobin concentration of test group A1 was compared with the disease control group. And our sample, that is mainly from soy yogurt, could be considered as a predominant of the rest of the test groups for resolving the deleterious effects of antibiotic associated diarrhea as seen from the table. Nextly, we have done this histopathological sample analysis of the colon. You can see the samples from 6 groups as shown, like normal control, disease control, standard, A1 containing the soy yogurt, A2 containing the mix of the 2 bacteria, and A3 also mix of the other 2 lactobacillus bacteria. So in the histopathological study, all animals from the normal control showed the normal layers of mucosa, submucosa, and muscularis, while disease control showed typical changes with diarrheal condition like inflammatory cell infiltration, you can see from the image. And test group A1, which fed with the fermented soy yogurt, did not show any abnormal changes, which could indicate it could work as antidiarrheal drug, shown from the histological analysis. Nextly, we have done the histological analysis of liver. Similarly, compared with the normal group, test A1 group fed with the soy yogurt did not reveal any specific lesions, and it was at par with the normal control group, indicating it could very well work as antibiotic drug from the results of the histopathological analysis in the liver as well. So these are the cecal. We have done the cecum samples. We have checked for the short chain fatty acids quantification from the antibiotic associated diarrhea animal models, and the cecal concentrations of lactate, acetate, and propionate was significantly higher in the rats of test group A1 fed with the yogurt compared to the rest of the test groups. So these were the just for comparative analysis, I've showed the test group A1 having functional soy yogurt, production of lactic, acetate, and propionate, and similarly the lower production of lactate, propionate in group in the disease control group. So to conclude my talk, the research can really identify the potential probiotic microorganisms which could be used as starter isolates and develop new functional fermented foods of northeastern region, as well as the rest of the country parts. And this lactobacillus strains producing B vitamins, organic acids, and soy glucans could be used for food formulation programs. And being popular for the pure vegetarian and the lactose intolerant population with cholesterol problems, this particular product could be taken forward due to its rich biofunctional and probiotic potential. And moreover, the treatment with functional soy yogurt contributed in the maintenance of the structure of the rat mucosal microbiota, as seen in the results from the animal models, in comparison with the normal group and disease control group, providing it to be an efficient drug in inhibition of this antibiotic acid diarrhea. So these are a few of the papers which were published during my course of time. I would like to thank Department of Dairy Microbiology, SMC College of Dairy Science, Raman Bhai Patel College of Pharmacy, Charusat, for allowing us to carry on the animal trials, Bioinformatics Infrastructure Facility Center, Tura Campus, Meghalaya, and Department of Biotechnology, Ministry of Science and Technology. Thank you. So we should support bacteria. That's the only culture some people have. That's how my talk ends over here. I tried to conclude in a very short time. Thank you so much for your time. If any discussions, please let me know.
Thank you, Dr. Sujit. Very nice presentation. Thank you very much. Anyone, if you have any questions, it is the time to ask.
I'm also thankful to Dr. Keyur Patel. I'm seeing in the group. Thanks to him also, because during my stay in Gujarat, he was the one who actually helped me a lot in Dr. J.B. Prajapati's lab. So I'm really thankful to him for the continued guidance and support. Thank you, sir.
Glad to know. Oh, yes, Prakash, sir, please.
Thank you very much, sir. This presentation, Sujit.
Thank you, sir.
And very good to know that you are working with BIRAC. In fact, we are actually submitting one other proposal to BIRAC.
Okay.
To the probiotics one day. Invariably becomes the experts or the funding agency these days with reference to the probiotic usage in Indian context.
Yes.
Nevertheless, what was your basis of selecting those cultures initially? That was one, and another one was when you incorporate in soya, how was their survival? First is the growth and survival.
Yes, sir.
And then third, whether you really called yogurt, soya yogurt, or only the fermented soya.
Okay, sir.
Just whatever, because I work with the NDR, Anand Agriculture University, so you know most
Yes.
of these things. Yeah,
Yes.
right.
So, sir, to answer your first question, like a basis of your selection on what basis you have selected.
Exactly.
Yeah. So, sir, basically the lactobacillus, like on the general criteria based on this catalase and gram-positive reactions, these are the basic things. But after that, we perform this biochemical analysis using this API kit. So,
Okay.
based on that, again, we selected the cultures, and we have done this gene amplification followed by the sequencing. So
Okay.
after that, we got confirmed that which particular lactobacillus, because after sequencing, many we have rejected because they are
Yes.
coming on this lactobacillus group. So we
Hello.
rejected them. So we rejected them and we took them forward.
Hello.
Yes, sorry, Sujit sir, please continue. Everyone, please keep yourself muted.
So after that, we have taken this forward based on the sequencing results, actually, sir. And to the next question, sir, you asked is.
Just before you go to the second question, but what was the selection? No, sequencing will give whatever the strain or culture identity.
Yes.
But for any some functionality with reference to some enzyme specific
Yes,
markers.
yes. Initially, we have done this, the technological attributes, like in the slide I've showed, like alpha galactosidase, beta glucosidase, and its polysaccharide production. Based on these three parameters, we chose those lactobacillus actually.
Wonderful.
Because lactobacillus could grow due to the presence of alpha galactosidase only in soy milk.
Yeah, exactly. Yeah, that is what actually I thought should be the emphasis should be given on that.
So based on that, I have taken the probiotic attributes for further.
Wonderful.
Okay. And
next.
So your next question was like the growth in soya milk you have asked.
Exactly. Because we are also fermenting soya milk.
Yeah.
Using some features, and what we did was actually we added something like a bacillus, which
Okay.
has a more protein.
Mm-hmm.
So what was your experience with reference to the growth of lactic? Because
actually, because soya milk is devoid of lactose, it's quite difficult to ferment in soya milk.
Yeah,
so that is the reason we thought of choosing this prebiotic inulin we have included in the study.
Okay.
Without that, it was difficult to, although it was fermenting, but it was like the coagulation was not intact, just like we do in skim milk and other milk. So based on that, we use probiotics so that at least we could give that proper texture. It should come. Yeah.
Yes. We are also actually looking for a co-culturing with others. And one of actually in case we actually added the bacillus along with the lactics. And then it was quite okay.
And yes, and the co-culture is the best option because alone it's difficult. If you use at least 4 or 5 cultures together, then it might work based on their rich enzyme activity. Yeah.
Get still many flavor or it was reduced considerably?
Sorry, can you please repeat?
Any flavor which we get, no, some sort of undesirable.
Yes, that beanie flavor. That is the reason while sensory evaluation, even in like I've done in Anand as well as in Meghalaya, people didn't like initially because of that beanie flavor. That
antibiotic laga panul chestunaru microbiology vaadu. Maaku telusu. Idi nee muddilo telusule.
Please go ahead.
Yeah.
Hello.
So, Bogle sir, please.
Can I interrupt?
Please.
This is Dr. Bogle, and see, I have very little work with soya. But then, what was the role of anti-nutrients in the growth of your lactobacillus? Because soya has a lot of anti-nutrients like beta-conglysinin, glycinin.
Yes. Like
how did you address them? I mean, yeah.
Anti-nutrients in the sense you have seen, I think in the slide I have mentioned regarding this polyphenol activity. So it was able to reduce the polyphenol in our biofunctional.
No, I'm talking specifically with respect to beta-conglysinin.
No, sir, that specifically that particular this conglysins we have not done it actually.
No, because beta-conglysinin is in one way responsible to give you that tangy flavor, and it has a allergen activity.
Yes, yes, yes.
So
Just to address, to connect the questions of Dr. Halami, he asked the last question where I was interrupted. I was unable to answer that. So
yeah,
that
I'm in line with Dr. Halami.
yeah, same question, sir. I'll address this. Like
Yeah.
the consumption
Yes,
of soya products is basically
Jain sir also subsequently.
yes. So it is
Please.
limited due to this objectionable beanie flavor, which most of the people doesn't like. And like you're
Yeah,
saying,
yeah.
and anti-nutrients, these are the responsible factors for causing flatulence or bloating if consumed in
Yeah.
large amounts. So
No,
that is the
because in one of the cases we have addressed this anti-nutrients
okay.
by using the consortium of bacillus strains, as rightly put by Dr. Halami.
Yes.
So I agree with Dr. Prakash Halami that it is always advisable to use a consortium which would take care of anti-nutrients and probably remove the tangy flavor.
Correct. In our
milk.
case, we use these organisms to check whether they can hydrolyze the oligosaccharides, which are responsible for this beanie flavor. This particular
See, hydrolyzing stachys by lactobacillus is a difficult phenomenon.
yes.
Yeah, I don't know how you have addressed it, how you have measured it also.
No, not stachys. We have done this alpha galactose. I'm talking about the alpha galactose activity.
See, you know, soybean contains a lot of stachys.
Yes.
Yeah. So if you address that, the protein content should go up.
Correct. And to address this also, we have done this that whether our yogurt is.
I think Dr. Halami has done a huge work on this. I think he'll be able to throw a lot of light.
Dr. Halami has been a big inspiration during my research time. I followed a lot of his papers while doing my work actually. So, and to address our concern, we have done this in vivo studies using the animal models.
See, what we are actually addressing is soya, especially to remove the anti-nutrients by solid state fermentation.
Correct, sir.
I don't know. I mean, even soya milk also would have those anti-nutrients, right?
Yeah, it has, sir. It has. But the particular conglycine you're talking, we have not done that particular work.
I mean, or you can have the protease inhibitors. There are a lot trypsin inhibitor, to be very specific.
Correct. Yeah,
I don't know. Sorry to have interrupted you. This is not my field of activity.
No, it's okay, sir. This is a suggestion. This is obviously a suggestion from me because whoever is right now, they're working in my lab, I can suggest them on this. Those who are working.
Since this is a open forum, I thought I would just interrupt.
Sure, sir. Please. Suggestions are always welcome to make the research perfect. Suggestions are really thank you, sir.
Intentions to disturb anybody's sentiments or work or anything. I mean.
No, sir. Suggestions are really, really welcome. I'm really appreciating this because this is the way we grow and learn. So this is very much important. So I can guide the other people who are working, taking my product forward. I can tell them.
Dr. Prakash would appreciate this. Am I right, sir?
Definitely, sir. Definitely. This is actually a very good occasion and platform also to share and then see how best actually we can utilize this unexplored soya. Definitely.
Yes. No, soya has already been explored worldwide, and now I'm happy to share that in India we have a largest of 50 tons solid state fermentation per day to address anti-nutrients like trypsin inhibitors, glycinin and beta-conglycinin, whose removal is to the extent of 90% plus.
Yes.
I have one question. Can I ask?
Please, sir. Please.
Dr. Sujit, do the lactobacilli produce some lipopeptides?
Yes, peptide study. Yes, they obviously but
lipopeptides.
Lipopeptides. No, so we have not done many trials on that.
So in the literature, have you seen?
I don't think they have addressed this. I don't think they have addressed this issue.
No. We haven't.
What I'm trying to say, from the literature, could you find out that lactobacillus makes some lipopeptides?
Okay, sir. I have to check this.
Does it make some lipopeptides?
Okay.
And biosurfactants, does it make?
Okay, sir. I'll check this.
Is a probiotic. It's a very, very candidate for probiotics, and it makes biosurfactants, which are lipopeptides. Okay, like surfactant, and it makes rhamnolipid like Pseudomonas.
Mhm.
Lactobacillus also makes biosurfactants.
Right.
And the biosurfactants of Lactobacillus are very acceptable
Correct.
because they are non-toxic, number
Yes,
one,
yes.
because it itself is a probiotic.
Mhm.
So when you get a chance, you can also explore this field, the biosurfactant,
Sure,
and
because
get a lot of ideas, to go
right,
about.
sir, right. Thank you. So this suggestion is very noteworthy, and like you've correctly mentioned, the cyclic lipopeptides, like they're really one potent class
heptapeptide.
of amphipathic natural biosurfactant.
The
Which get-
heptapeptide, like
Yes,
surfactant, it is having 7 amino acids.
correct.
Okay. So which is the best part of your work, you think?
Specifically, first thing is to interact with the tribal region out there. Since you have been in Northeast, you know the situations over there. It's quite difficult because they don't share their secrets, how they do the make the fermentation, how they use. They're consuming the microflora unknowingly. They make a lot of amount of fermented foods, fermented bamboo shoots, fermented meat, fermented like rice beer, but they're consuming probiotic microorganisms with unknowingly, and along with that, because it's prepared in some unhygienic way, along with some good bacteria, some pathogenic bacteria is also there. So when I isolated the lactobacillus from those fermented foods and beverages, I found that actually. So first thing was to interact and to let them know how they can make their product more hygienic. If they can get a starter culture from our lab, which we have handed them over, so that they can use that in their own local business.
Okay.
That is one thing.
Doctor, do you have biodiversity approval for this?
We have taken regulatory approvals from the government of Meghalaya.
No, biodiversity board.
No, sir. We didn't get for this.
Theoretically, you should obtain that. No? You're talking of commercialization, certain things.
No, we haven't done commercialization yet. I'm not.
No, but then before that, you need to have approval from biodiversity board.
Correct.
I don't know. This is just a part of suggestion that if you want to commercialize, kindly approach biodiversity board for approval.
Sure, sir.
Am I right, Dr. Prakash?
Yes, sir, definitely.
Correct. I have one more question.
Yes.
I think in one of our interactions, we were talking about biodiversity board approval for such things. I mean,
exactly.
When he does commercialization, he will do it. Right now, he is not commercializing.
No. I'm sorry. Your value for the product would go up if you have the approval.
Right, sir. Right.
All right.
You said that I'll sell it to somebody and he will approve it's his ball game. Nobody is going to do that.
Yeah, he can get. Try to get Sujit.
Sure, sir.
Chennai. The NBA,
actually, you should have got this approval before you started your PhD work.
Okay, but our intention was not to commercialize. We didn't knew that this was going to work to how level. Because
you are talking of commercialization.
No. I'm just saying commercialization. We have like the next people who are working. Even I'm guiding because I'm in BIRAC. I know this is our main target to bring that.
So if you are in BIRAC, kindly advise them.
Exactly. That's what I'm saying, sir.
Biodiversity board approval first.
Yes. Even while the project comes to us, we suggest for different regulatory approvals taken project to move forward. So since
I think in one of the proposals we were asking Dr. Halami also that do you have biodiversity board approval?
Correct. So now since my target was not commercialization, there was none of my objectives are commercialization. So now when the project,
I mean, being associated with BIRAC, this and that, don't you think that any research you
obviously now correct, sir,
should bring a value addition to the table?
Obviously, since I'm in here, since I'm knowing how to do it, obviously I can guide my people how they can take this forward to reach the people. So that the technology should not be in lab; it should go outside also by hand-holding with some industrial collaboration and taking the necessary approvals.
Thank you, Bogle.
Otherwise, your research is of no value.
Correct, sir. That's
I think Swarnajit sir has a question. Swarnajit sir, please.
One question. Sujit, have you preserved your cultures, the important cultures which you isolated?
Yes, sir. It is still, they're working. Juniors.
How did you preserve them?
So it's in -80 glycerol stocks. It's
very good. Right? That's a very good way. Yeah. I can see Dr. Neena Mehta. Dr. Neena.
Anji sir, namaskar.
How are you?
Okay, sir, I'm fine.
You can ask some question to Sujit.
I just wanted a general question for common public, although this one is for research purpose. that those who are allergic to milk, so the most common yogurt that can be produced from soya milk at home. So how
Yes.
will you help those people?
Yeah, for that,
I mean,
that is the
is
reason.
it easy to make yogurt with soya milk with lactobacillus?
Yes, yes, absolutely. You can make that in home also. You can make that.
That you can give some reference for that, because it is not very commonly available in all general stores like
Yes.
the milk yogurt, right?
Yes, correct, correct.
So I would like to know the reference as well as to make it more common to public so that people can make use of this.
Sure, sure, sure. Ma'am, you can reach out to me. I can give you with some.
Like the tofu has become very common in
Yes.
market.
In
It is available everywhere.
yeah, in India also you'll see a lot of
product.
coming out
But
like SoFit.
milk, soya yogurt is not very common that you can go anywhere and get it.
Yeah, it is not available right now. People are still to accept it,
Yeah.
but SoFit they are producing soya milk and all. You'll get in all the outlets. SoFit, SOFIT.
Yeah. So just help. I'm one of the subject.
Sure,
So,
sure. It will be like I will be happy to help. You can take my ID from
Yeah.
Mr. Jai. Then you can contact me personally so that I can guide you.
Mhm.
Sujit sir, I have one request. Why
Yes.
don't you type your email ID in the chat box? Someone
Sure.
else also requesting me. So anybody who have questions, they can direct it to you.
I've written in the chat box.
Please.
So any queries you can reach out to me for further clarifications or if you need any help.
Rakshak, do you want to ask
Yes,
any
sir.
question?
Hello, Dr. Sujit.
Yes.
I am Dr. Rakshak from CSIR IHBT Palampur. I have completed my PhD from Northeastern Hill University, Shillong.
Okay.
And I must thank you for this comprehensive study. Although we also tried extracting, and although my work was on environmental microbiology, but some of our research paper are on probiotics from traditional food of Meghalaya and other northeastern states.
Yes.
But we lack this kind of comprehensive study where you have done animal model study and have given a true picture that how important it is to exploit or explore the traditional knowledge of Northeast. So first of all, I would like to congratulate you. Very
Thank
wonderful.
you. Thank you.
And like as you are in BIRAC now, we also try like